And the central dogma, a dogma is an established process, right? And it's just the way it is, like it's a doctrine, if you will. Okay, it's what we say. This is how it is, period. And this is how it is, period, for every living thing we know. That in terms of how genes are expressed to produce a product, it occurs this way for every gene, in every cell, in every living thing on the planet. Okay, so I would say that's dogmatic. And it kind of just goes like this. You have a piece of DNA, right? And from that piece of DNA, there are little exceptions here that we could talk about, but you make a piece of RNA. And we're going to call, for sake of a longer discussion, we're going to call that piece of RNA messenger RNA. There are other kinds of RNA that can be produced that do other things. I'm looking at the bigger picture of trying to produce a protein, and the product of this expression of DNA here is messenger RNA, because it's the messenger RNA that is then going to be used to, I'm just going to say here, to help us produce a polypeptide. Now, this is a language, right, of A's, G's, C's, and T's. So this is a nucleotide language. Right? This also is a nucleotide language, but it's a language of A, G's, C's, and U's. Same kind of thing, right? These are still nucleotides. Chemically, they're identical to, they're almost identical to each other chemically. To protect the DNA, to protect the genetic information, DNA happens to be double-stranded. That's just for protection. Okay. To use the nucleic acid, we have to make it single-stranded. That's why we use RNA instead of DNA. It doesn't have that kind of double helix appearance to it. But this is just taking one language and writing it down again. And that's called transcribing. Right? When you transcribe something, you just write it down. So that's all we've done there. So that's why this process is called transcription, is because you don't change the language, right? You just do it. Now, it's a complex process. It involves a variety of enzymes called polymerases and things like that, and certain conditions that have to be appropriate. We have to identify these promoter regions and all of that. From a genetics biology point of view, if you take more advanced biology classes or genetics classes, you'll learn about, we could spend, I used to teach genetics, and we could spend, a couple weeks just talking about transcription, because it's a very, involved process. But for us, it's beyond what we need to really care about. Okay? But it's not real simple by any means. Where this occurs has to be where the DNA is, right? And where is the DNA? It's in the nucleus, right? So this process of transcription has to occur in the nucleus. So all of the enzymes, all of the materials, all of the things that we need to do transcription must occur within the nucleus to allow this process to occur. And the whole purpose of the nucleus, okay, maybe that's a lie. Not the whole purpose, but a main reason why eukaryotic cells evolved the nucleus is simply to protect the DNA. When you take and some of you have had microbiology. When you take microbiology, you'll study, so prokaryotic cells don't have a nucleus, right? So their DNA is more vulnerable. But their DNA, the chromosomes, are not linear. They exist as a covalently closed circle. So they're protected by themselves because they're circular. Nothing can come to them from the ends, right? Eukaryotes, on the other hand, their chromosomes are kind of broken, and they're linear. And there are these things called exonucleases out there that like to just chew up DNA from the ends. Okay? So to protect these linear pieces of DNA from these exonucleases, a lot of viruses are exonucleases and things like that, we store them in a structure, a compartment called the nucleus, and that protects them. So that's why they're protecting. But what makes protein, if you remember from studying organelles, are ribosomes. And ribosomes provide a location for that. They're not in the nucleus. So the whole point of transcription is to just take a copy that's single-stranded out of the nucleus and take it to the cytoplasm, or rough ER, right, where the machinery is that can do this process here, which is going not from a language of nucleotides, but from a language of nucleotides to a language of amino acids, right? So that's like English to Spanish or something, right? So to do that, you need to translate the message. So that's why this process is called translation. It's simply because the chemistry language is very different. This is a nucleic acid nucleotide message, chemically incredibly different than polypeptides that are polymers of amino acids. Now, this also is a process that we could spend a lot of time,